Hello children, this is grade 10 biology and today's topic is heredity. In this chapter we are going to learn about heredity, Mendel's contribution, laws for inheritance and sex determination. So, here we are going to study the mechanism by which variations are created and inherited. Genetics is the branch of science that deals with heredity and variation. Variation is the difference in the characteristic features among the individuals in a species, that is, for example, the color of the eye, the height of a person or the color of the hair. Whereas heredity is the phenomenon of inheritance of traits or features from parents to offsprings, that is their progeny. Now let's move on to the next topic, that is accumulation of variation during reproduction. We have learnt in our previous chapter about asexual reproduction and sexual reproduction, where in asexual reproduction the chances of variation are less because the daughter cells or the offsprings are formed from a single parent, whereas sexual reproduction the variations are more because here there is an involvement of two different organisms having different DNA. These variations occur due to error in DNA copying. Now let us learn about variation with the help of an example. Let us take bacteria as an example which undergoes asexual reproduction. From the figure we can see the organism at the top that gives rise to two offsprings having slight variation. When they produce offsprings, 4 offsprings, we can see some new variations in the offspring, but most of the features are similar to their parents. This is the basic phenomenon or the concept of heredity. These variations are very important for the survival of organisms. It gives survival advantage to the organisms in most of the times and also help them to cope with the new changes. But variations are not always beneficial and do not give equal chances of survival and can sometimes get eliminated. Now let us learn about Mendel's contribution towards the rules for inheritance of traits. Gregor John Mendel discovered the fundamental law of inheritance. He stated that genes come in pairs and are inherited as distinct units, one from each parent, that is from the maternal and the paternal DNA. He tracked the appearance of these traits in the offspring as dominant and recessive gene. Recessive means the one which gets suppressed in the offspring. For these experiments he chose pea plants because of their visible traits with the contrasting characteristics and also because of their shorter lifespan. Mendel in the first experiment, he experimented with one characteristic feature at a time. For example, he took tall and short pea plants. When he cross-pollinated the tall plants with the short plant, he found out that all the offsprings were tall. These offsprings are called as the F1 generation or the F1 progeny. Mendel wondered what happened to the short characteristic feature. Did they disappear? Then the second generation also should have all tall plants. So to test this prediction, he self-pollinated the first generation, that is F1 generation. To his surprise, he found out that 3/4 of them were tall plants and 1/4 of the offsprings were short in the F2 generation. This means that the offsprings reflect only the dominant traits from one of the parents. Dominant traits are observed as 3:1 ratio to recessive trait, that is 3 are tall and 1 short. This is called the phenotype ratio, whereas in genotype ratio they are in 1:2:1, that is 1 purely tall, 2 mixed and 1 purely short. In this explanation, both capital T and capital T small t are tall plants, while only small t is a short plant. This means the presence of one dominant trait is enough to make the trait prominent or visible in the offspring. Dominant traits are denoted with a capital letter, whereas the recessive trait is denoted using a small letter. Thereby, we can say 2 copies of the recessive trait is required to be prominent or visible in the offspring. Same with violet and white flowers, where violet is the dominant trait. We get all violet flowers in the F1 progeny. When F1 progeny is self-pollinated, we get the result in 3:1 ratio, where 3 are violet flowers and 1 white flower. The result of these experiments led to Mendel's first law of inheritance called the law of segregation. Now let us see the second law. In Mendel's second set of experiments, he experimented with 2 traits at a time. The result of this experiment led to Mendel's second law of inheritance called the law of independent assortment. For example, in this parent generation, yellow and round pea is crossed with green and wrinkled pea. In the F1 generation, all are yellow and round. When these 2 hybrids were self-pollinated, F2 generation is formed. In the F2 generation, 9 out of the 16 were found to be round yellow, 3 out of the 16 were found to be wrinkled yellow, 3 out of 16 were found to be round green, and 1 out of the 16 was wrinkled green seeds. That is, they were in the ratio of 9:3:3:1, where 9 were dominant, then round green and wrinkled yellow, that is 3:3 ratio, were mixed varieties, and 1 was purely recessive, that is wrinkled green. This experiment demonstrates that gifts for each character are independent of each other. Now let us see how these traits get expressed. We have seen that in the first and second set of experiments, that traits are inherited from the parents to their offsprings. But how does it take place? We know genes are responsible for the transfer of the traits from the parents to their progeny. For every characteristic in an organism, there is a dominant gene and a recessive gene. Each parent possesses a pair of genes for each characteristic feature. Genes are located in the chromosomes of a cell. Let us take the example of tall and short trait. So, the parent can possess dominant gene capital T and recessive gene small t, or both dominant genes, that is capital T or both recessive genes, that is small t. There can be any combination of these 3. So, during sexual reproduction, as gametes are haploid, they possess any one gene out of the 2. That is, one gamete from the male parent and one gamete from the female parent fuses, and the fused cell decides whether the progeny will be tall or short, depending upon the dominant and the recessive gene inherited by the offspring. The next topic is sex determination. Let us learn how the sex of a newborn child is determined. In lower animals, sex determination is based on environmental factors and not the genes, like the temperature in which the animal's egg hatch decides whether it would be a male or a female. Whereas in lower organisms like snails, the organism can change their sex during their lifetime. Coming to humans. In human beings, sex determination depends on the genes. Each parent possesses 2 sets of genes. Now, during sexual reproduction, only one set of gene is possessed by the gametes, that is, one from the male gamete and one from the female gamete. Men possess XY chromosomes and women possess XX chromosomes. So, during sexual reproduction, mother always contributes X chromosomes, and if the child inherits X chromosome from the father also, then the sex of the child will be female, and if the child inherits Y chromosome from the father, then the child will be a male. So, the sex of the child is determined by what they inherit from their father and not their mother. So, in this way the sex determination takes place in humans.